Welcome, everybody, to today's coffee chat. We will be talking about drug hunters' capabilities today. And we have the entire team here today. So I'm pleased to be joined by our scientific director, Lou Pennington, by our senior editor, Hesse, by our editor, Rory, and by our scientific analyst, Shaima. So welcome, everybody, to the show. It's a pleasure talking to you. I feel like we're in a live editorial meeting here, and it's going to be a very fun time to talk about Drug Hunter. So yeah, for all of you who are dialed in, so this meeting is going to be recorded, and you can ask your questions in the Q&A box. Also, feel free to use the chat box to say hi and say where you're dialing in from. But yeah, if you later on want to rewatch any of those things that we covered and talked about, the video will also be on YouTube next week for you to stream. So yeah, with that, we get started today, and we all are going to cover different features of Drug Hunter today. And we're going to start with Lou. I will start sharing my screen, and then Lou will walk us through a case study and talk a little bit about how we select those case studies. And I will hide my meeting controls here so you won't see those black bars. All right, Lou, floor is open for you.
Oh, cool. Yeah, the last black bar disappeared. Thanks, Dennis. So yeah, as Dennis mentioned today, each one of us is going to be giving us a slightly different presentation. So you're going to get, I think, a pretty nice overview of what sort of content and what sort of capabilities a Drug Hunter membership can afford you. So I've been asked to briefly present one case study today to really try to give you a sense of how our case studies are crafted, how they're structured, how we like to make sure that the transferable insights are highlighted, as well as how you can leverage the reference hyperlinks that are embedded into the articles to get more detailed information when you desire to do so. And then hopefully, this brief discussion on this case study will also give you a sense of how you might be able to use our case studies as the basis for journal club discussions at your own companies. So today I've decided to focus on Vividian's Werner helicase inhibitor, VVD214. This is a really fascinating molecule and a really fascinating drug discovery story that we've covered in a number of different contexts here at Drug Hunter. And we do that because it is so fascinating, all the way from the therapeutic hypothesis to the pharmacological profile of the molecule. So Dennis, if you scroll up just a little bit, first I'd like to kind of show people how our case studies are constructed, because we might have some people listening who aren't all that familiar. So, obviously first we have the title and the name of the molecule, and you get a sense of what that molecule is like. So here we describe it very briefly as an oral covalent allosteric inhibitor of Werner helicase. We give you a sense of where it's at in discovery or development and what it's being targeted towards in terms of disease indications, the origin of the chemical matter, if we have access to that information, as well as some of the key ways that the molecule was optimized. And then we also like to include the source for the article. So you can see here, this article actually stemmed from a presentation at the AACR meeting in 2024, the New Drugs on the Horizon. So this is something you could not get from the literature. This is something that was presented at a meeting. We had people present taking notes, and we used those notes as the basis for this article. So just to start off from top to bottom, so you can see underneath that sort of introductory information, we have the highlights for the article. So this allows people to very quickly get a sense of what the most important parts of the article are, especially what we call these transferable insights. So a lot of times our articles might be read by people who aren't working on this therapeutic target or even this therapeutic area. But we believe there's a lot of generalizable lessons and transferable insights that can be harvested from one drug discovery program to the other. And so we try to highlight those in this part of the article. So if you scroll down a little bit more to the introduction, so I'm not going to go deep into the introduction, but what we try to do at the introduction is really try to give people a sense of the content of the article, the context of the drug discovery effort, as well as the importance of the work. So we try to make sure people understand how this drug discovery story fits in to the broader drug discovery world. And so included here, you can see in figure 1 for this introduction, not only do we include the molecule of interest for the article, but also other molecules that are in this space. And in this instance, we highlight HRO 761. This is a Werner helicase inhibitor from Novartis. So we want to make sure people are aware of all of the important things going on in these arenas. And so what was the therapeutic hypothesis for this program? What was the goal? So the goal for Vividion was to really try to leverage this concept of synthetic lethality for treating cancers that exhibit high microsatellite instability from deficient DNA mismatch repair. And specifically, they wanted to develop inhibitors of Werner helicase. And so in this figure that is shown here, you sort of have 4 different scenarios where you can have mismatch repair proficient or deficient cell types. These could be cancer cell types or healthy cell types. And then you can have Werner helicase. You can have active or absent Werner helicase, for example. And there's sort of 4 different scenarios. I don't have time to go into all of them, but for the purpose of this therapeutic hypothesis in this story, the hypothesis was that if you have mismatch repair deficient cancer cell types and you could pharmacologically knock out Werner helicase, you could treat tumor types that have high microsatellite instability and leverage synthetic lethality. So the absence of mismatch repair mechanisms coupled with the pharmacological knockout of Werner helicase could lead to synthetic lethality. So that was the hypothesis of the program. So if you scroll down, the way that Vividion went about starting this program, because as people know, helicases are pretty difficult targets to drug. And so Vividion's developed this really wonderful and broadly applicable chemoproteomics platform that they've used to identify chemical matter for a number of interesting targets, including not just Werner helicase, but also KEAP1. And we've discussed those drug discovery stories on Drug Hunter. So really just quickly kind of going through this here in figure 3, you can see the way they go about doing this is they treat either cell lysate or live cells or even live animals with their compounds of interest or covalent compounds. And then that's followed by treating with the probe molecule, digesting with trypsin, and then enriching those probe labeled peptides. And then they're able to quantify the peptide signals using LC mass spec. And this allows them to simultaneously assess potency and selectivity, and also results in some serendipitous discoveries. So in this case, they screened 10,000 compounds against 300 proteins. And what they identified, if you scroll down, was a series of compounds exemplified by 109063 that bound to cysteine 727 of Werner helicase. And as they launched their optimization campaign, starting from 109063, what they found is simply by swapping out the methyl group with a phenyl group, adding 2 nitrogen atoms to the phenyl ring, and then adding a methyl group to that vinyl sulfone moiety, they actually went from a compound that was demonstrated to be an ATP competitive inhibitor of Werner helicase to a compound that was found to be ATP cooperative. How did they find that? They saw that this compound actually had greater potency in cells than it did in cell lysate. So they decided to capitalize on this serendipitous finding and ultimately transformed 127101 to 133214 that we call VVD214. This is the clinical compound. So really a nice piece of work capitalizing on a serendipitous pharmacological observation. So if you scroll down a little bit more, what they found is that 214 covalently engages cysteine 727, stabilizes a closed state conformation, and inhibits the ATPase activity of Werner helicase. I don't have time to go into detail now. Hopefully our members can dig into this a little bit more if they wish, but if you scroll down a little bit, Dennis, a little bit further, So what they found with this compound is that it was active. It showed growth inhibition activity in cell types with high microsatellite instability due to deficient mismatch repair mechanisms, but showed no activity in cell lines, cancer cell lines, that didn't have this sort of characteristic. If you scroll down a little bit more, this cellular activity was recapitulated in patient-derived xenograft models. So you can see that 214 showed tumor regression activity in these PDX models that that had high microsatellite instability, but were not active in PDX models that did not display that cell types that have microsatellite stability. They could even do a mutational study if they mutated out that cysteine 727 to an alanine. Then the 214 was not active. So further sort of confirmation of that pharmacological hypothesis that they had on the program. So then what's next? So if you click on that link there, we provide information for the clinical trial. So you go right to clinicaltrials.gov. And if you really want a lot of information about how they're testing this molecule in the clinic, you can get that if you desire. And then if you toggle back to the case study, Dennis, we always like to include the medicinal chemistry synthesis of the molecule if we can get our hands on that. So we'll go through the patent applications and look to see if we can identify, you know, that clinical compound and the chemical synthesis of that. So if you're interested in that, you can dive a little bit deeper. We do provide that sort of brief summary of the chemical synthesis. And then if you scroll down a little bit more, we always like to include all the relevant patent documentation for the article as well. So that's all I have in my 5 or 7 minutes today. There's a lot here in this article. So it's pretty dense. So I encourage people, if you have a membership, to go in there and read it in detail. And if you don't have a membership, get one. And I think you'll enjoy the content on Drug Hunter.
Awesome. Yeah. Thank you so much, Lou, for walking us through this case study. It's super interesting. Yeah. Shaima, would you like to talk a little bit more about how we cover conferences and maybe give an example of a case study that we recently covered for a conference?
Yeah, sure. Hi, Dennis, and hi, everyone. Thanks for joining us for the coffee chat today. So I'm going to talk about conferences that we cover and the benefits for member. So at Drug Hunter, we cover key industry conferences where new discoveries and first-time disclosures are presented. And to give you an idea, we cover conferences like the ACS national meetings, the AACR, the ACS medicinal chemistry, RSC, and the International Symposium on Medicinal Chemistry. So these events contain details you won't find in the traditional peer-reviewed journals, such as specific tox findings. An example is one of our latest first-time disclosures, which is about the NLRP3 inhibitor AZD4144 from the EFMC ISMC in Rome that I will briefly share with you today. So actually, like the structure of the first time disclosure article is not very different from what Lou has just presented in a case study, but sometimes we tend to do briefs when we already cover that target and we already have covered the competitive landscape and the process of target and target validation and why it's important. And this is why AZD4144 is in a brief format. And again, the same structure. The highlights is just giving the reader the transferable insights and what are the most important, I would say, conclusion or take-home messages from the article. And we definitely make sure that we cover these insights for our readers from the presentations and talks that we attend at the conferences. So when it comes to the introduction, again, it's the same concept as the case study, and but it's like a little bit on the brief or shorter side, especially for NLRP3. This target is really hot, and we already covered at least 5 molecules in either case study or brief format. not to mention the patent highlights and the molecule of the month. So yeah, Dennis, thank you for clicking on the link. This takes you to the drug hunter search and all the NLRP3 coverage that we have done before. So you'll find everything you need to get an idea about the competitive landscape and what has been done for NLRP3. So NLRP3 inflammasome is absolutely of a high interest target now. And there are multiple companies that are advancing compounds into clinical trials that hold potential to treat a broad range of inflammatory and autoimmune conditions such as IBD, lupus, Alzheimer's disease, and others. The interesting thing about AZD4144 is that it was one of the first compounds that diverged from the hexahydroindocene that was seen with the prototypical molecule from Pfizer, and then many companies came after Pfizer and they have developed drugs or investigational drugs carrying the same scaffold. So we also make sure to cover the hit identification and process in details, what technologies the company has used, what assays also they have applied during the hit identification process, and any relevant biological information that the reader might need to understand the story of hit identification. For example, in AZD4144, for example, they have used the IL-1 beta assay coupled with the SPEC assay. So again, as Lou said, I cannot go into like very fine details now, but here is where the story started from. If we look at figure 1, they have identified initial 12 hits that have the pyrrolotriazine core, and they had like an IC50 between 0.7 to 11 micromolar. They have done hit expansion studies, and they found that 300 compounds have the northern basic amine or basic amine at the upper side of the molecule. They have improved potency in LRP3 potency. So they were really interested in pursuing that basic amine series, and AZ1178 was of the very first compounds that they have identified. However, this molecule suffered from hERG inhibition, in vitro toxicity, and in vitro phospholipidosis. So more optimization was done around 1178. And here I like to always to highlight what parts of the molecule were changed for visual learners like myself. It's just easier to track how the SAR was done. Here we see that they have changed the pyrrolotriazine core into a phthalazine core. They have installed a hydroxyl group, and they have swapped the trifluoromethyl for a methyl sulfone. So they were able to improve many parameters: the potency, the in vitro safety, the hERG liability, even in vitro, or the molecule tested negative for in vitro phospholipidosis. When they advanced the molecule into 7-day dose range finding study in rats, they found that phospholipidosis or vacuolation, which is a mark of phospholipidosis, was found in vivo. So now the company has to like think again about the basic nitrogen, because there are some studies that showed that it could be implicated in phospholipidosis. So they revisited the head again, and they evaluated a neutral sided chain instead of the basic one. And through the optimization process, as you can see here, they were able to improve the potency. Then they were struggling in AZ111 with the hERG activity. It was like hERG inhibitor at 10 micromolar. Then they have done more optimization, and absolutely, like here, I'm just talking about the things that worked. Within the text, you can find more details about the things that did not work. When they did further optimization and cyclization of the upper part of the molecule, they were able to solve many liabilities, except now that they had the micronucleus formation in vivo, which is a marker for genotoxicity. So I feel like the scientists were like so fed up by that time. It's like they were facing the hERG, they were facing the phospholipidosis, and then they have the micronucleus formation. So they have like they have all of these assays at the front line, and they were testing their molecules that emerged from the optimization process for all of these liabilities early in the discovery process. So again, more optimization. They were able to discover AZD4144, which has like improved or proved or good NLRP3 potency, solubility. It was also the hERG inhibition was also solved in that molecule with other like safety parameters or safety concerns that were associated with the prior molecules. So I want you to imagine now having to write down all of this during a talk or a presentation. That would be really very time consuming and probably distracting for some people who like to be like fully engaged with the presenter all the time. So when like if you have a Drug Hunter membership, when you're attending a conference, you just don't worry about all this, taking notes of all of these details or doing any like research afterwards, because like shortly after the conference, we make sure to publish these first-time disclosures in as much details as the presenter presented at the talk. So now you can focus on listening, learning, networking with other people. And our goal is just to make sure that you have all the transferable insights from every first-time disclosure and non-first-time disclosure talks at the conferences. So if you scroll down, then it's also, we have the preclinical profile that made it or qualified that molecule to go further into development. It has a very clean in vitro safety profile, and it has like also a favorable PK profile. And then we also presented, or we have showed that in the NLRP3 inhibitor AZD414 progressed to clinical trials, and now it's in phase 1 trials. And here we also have the identifier number at clinicaltrials.gov website. So, and also again, this is what we do at Drug Hunter, is that we don't only provide the exact presentation or what the presenter has talked about during the presentation. We go beyond what was presented. And we have observed that it was like AstraZeneca was one of the first companies to diverge from the hexahydroendazine. And the interesting thing is that there were companies that like were doing similar scaffolds. So when I done a patent research on SciFinder, I found that Merck, Takeda, Kodiak Sciences, and PTC Therapeutics, they also started to work on phthalazine or phthalazine derivatives as NLRP3 inhibitors. So we always make sure to go beyond what only was presented at the conference by providing everything the reader needs to know about that particular topic. So yeah, at Drug Hunter, we aim to save you time, allowing you to stay informed without committing to extensive travel or follow-up research reports. Also, we know that you cannot attend every single conference, and even at a single conference, there could be like 2 parallel sessions. So we want to make sure that you don't miss out on anything. I mean, you can attend one session and Drug Hunter is covering the other session for you. Also, most companies, as we may know, they budget for only one conference per employee per year. So there are like many really interesting conferences and talks. So Drug Hunters make sure that scientists don't miss on the disclosures and the discoveries that are presented in conferences that they do not attend by Drug Hunter is having their back, and we're always there to provide them with all the fresh discoveries and insights from those conferences that they do not attend. So yeah, that's said. And if anyone has any question about conference coverage, please feel free to ask in the Q&A.
Awesome. Yeah, thank you so much, Shaima, for covering that. Yeah, it's such an interesting story that hexahydroindazine has been called the elephant in the room by many. And it's like really an interesting story here from AstraZeneca. And yeah, thanks for sharing how Drug Hunter can help you covering some conferences in the audience. Okay. Yeah, with that, we go to another feature that we have on the website. And I'd like to invite Matt to talk a little bit about the patent search tool and the patent highlights that we provide on Drug Hunter.
Thank you, Dennis. Yeah, I'm Senior Scientific Editor here at Drug Hunter, and I head up our patent search and patent highlights features, which our members say bring real value to them. I mean, one of the key things I think that Drug Hunter provides is, a lot of scientists, I think, wish they had more time to be able to go in depth into the literature. But even if they were given more time, I don't think many scientists would really enjoy, diving into the patent literature. It's not quite as engaging, and so we bring you what we think are really the curated highlights and the most important kind of IP disclosures in drug discovery. And so each month, what we do is we try to identify every drug discovery relevant patent that has been published. And we do use AI tools to do this, to identify it via companies, via targets, via chemical matter. But then I think what's really different, really valuable, is that we then have our scientists within the team actually triage that and manually read them. And we triage it down to about 200 to 250 of the really most important drug discovery patents that were published in the month. And what we do is we go through them, and we tag them obviously by the originator, what company's been working on them, what the targets are, what the therapeutic areas. What we also do is we do a manual annotation of what's contained in there. And typically we'll include the number of compounds that are covered, maybe how many different series. We'll look at the potency of it, and then really go into and try and find what would be the super interesting things that people would want to know about that patent. Is there PK data? Is there tox data? Is there formulation data? Right? Is this a crystal formulation one? Is this likely going to be their clinical candidate? And so each month we then release a table, a searchable table like this one shown here, which is text searchable, and we'll release this at the end of the month for the month previously, and then we'll collate that into this searchable database, which has all of our curated patents in it. And this is fully text searchable within the table, so it doesn't search the patents, but it will search the annotations, and it will allow you to see, okay, what's happened last month? What has Merck published, right? Who has published something in oncology? And it will really give you an ability to look outside of kind of the area that you're focusing on to give you a broader appreciation of what's going on in the industry. So you can use this text search, or you can also use the filter search. If you click on this purple Show Filters button, it will then give you kind of a tick box menu of kind of the targets, the MOA. So if you're interested in a specific target, you can click on that, say NLRP3, and then it will give you then who is working in NLRP3, who is published there. You can also, so if you click off that box there, Dennis, and then you can go down, say, to the company. So if you click on perhaps Novartis, this will then tell you, okay, what has Novartis published? And obviously you can do a search like this in SciFinder, and you're likely to get 1,000 results. Whereas what you really get with Drug Hunter is the curated, the most interesting things that you should know about that have been published by these different companies. You can also say, if you click on Roche as well, this will allow you to compare, okay, what are Roche and Novartis working? Where are they competing? And if you scroll up to the top on the left-hand side there, Dennis, what are the ones which they're both in? So if you click on NLRP3 here, it'll tell you, you'd be able to directly compare, okay, what are the patents that Novartis and Roche have published on this recently? And you can see, if you look at, say, the Novartis one here, you see their one published in February. This is one example, right? That probably means it's a very important molecule. And so right away, you've drilled down to really get to the really important knowledge. Whereas if you do a SciFinder Cortella search, you get 1,000 results. This allows you really quickly, in a user-friendly way, really get to what the key things that are going on. Additionally, what we do is each month as a team, we go through, we select what we think are the really important patents that have been published that we think people should know about. And so each month we'll also publish what we think our highlights are. We'll do a dive into each of these, give you a sense of why the target's important, what's the background, who are the other people in the areas, and what we really think is scientifically important. So if you go to, say, the one from August, Dennis, so the first one here is something I think is super interesting. So this is ACR1C3, and sometimes what piques our interest is, okay, have we not seen this target before? Why is Novartis interested in this target? And so this target is actually a lysyl tRNA synthetase, which is really central to ribosome protein synthesis. You can imagine this can be pretty toxic. And so what Novartis is actually doing is it's using this as a prodrug strategy in NRF2 activated cancers. Obviously, NRF2 is kind of a hot area right now. NRF2 is a transcription factor, and one of the target genes that it upregulates is a ketoreductase. So what they've done is they've published this chemical matter here, where you can see compound 40 is this ketone prodrug, where in NRF2 amplified cancers, where you have an upregulation of this ketoreductase, you get selective activation of this ketone to the alcohol, which is then the active inhibitor of the lysyl tRNA synthetase. And so you get cell selective activity. And this is the kind of really interesting science, really hot kind of things that we want to pick out and share with our members. Another pattern we picked out in this, if you scroll down, we also pick out the kind of data from each of these as well, and try and give you so straight away you can find the really important stuff that's in that pattern. So I want to share this one as well. So this is KRAS, and the top set of compounds here are from Revolution Medicines, which we've covered in depth with these really, really significant macrocycles, which are able to engage KRAS by kind of forming a protein-protein interaction with a ubiquitously expressed protein cells called cyclophilin A. And obviously, this has been kind of a really new approach to doing this. And you can see that Roche have published this pattern where they've taken that scaffold and they've added now this macrocycle here on the indole. And what this does actually is this gives it kind of benefits in terms of its clearance and its hERG activity. So you can see if the top two lines of this table here, you can see the hepatic clearance is reasonably high. Compound example 24 from this pattern and now has reduced metabolic clearance. And it's actually got a much bigger window with regards to its hERG liability. So again, these are the kind of things that we want to pick out and be able to share with our members with this. And we get really good feedback that this is actually really valuable because lots of people don't have time to be able to read through patents to find these kind of in-depth insights.
Awesome. Yeah, thanks for sharing that, Matt. I actually have to admit I use this like every day. It's so useful to find out what's hot right now. Like oftentimes in the peer-reviewed academic literature, you find things that companies deprioritized or worked on maybe 10 years ago. But the patent literature really has the gems, and I think the team does a really great job in extracting those. All right, I want to pass it on to Rory right now, and Rory is going to tell us a little bit more about the molecules of the month and how we select them. Rory.
All right, cool. Thanks so much, Dennis. Happy Friday, everyone. I'm super excited to be here with all of you to present kind of how we think about molecules of the month, what exactly is molecules of the month, and what you can expect to get out of it. So where Dennis is at right now, we actually have a landing page for molecules of the month. So every month this is, of course, updated. Actually, this morning we just published the October 2024 molecule of the month. So this weekend, feel free to take a look and read through and see what our selections were. But I don't want to give away the thunder for that article. So let's select, for example, the molecules of the month, September 2024. And so this was a really fascinating month. And so, what we provide for you guys are essentially these downloadable PDF posters of our top 10 molecules that we as a team selected. And we'll get into kind of the nitty-gritty about how we select these molecules. But this is a very nice way to get a feel for, okay, what is out there, what are companies working on, what are some structural inspirations that I can perhaps apply to my project. So we of course, for example, we give you the names of the molecules, the targets, the mechanism of action, the most recent clinical development status, and where these molecules appeared. So for example, if they appeared in the primary literature, we'll give those citations and then the companies that are associated with them. So if we scroll down a little bit, we give you more details about why these molecules are important. And so now in a text form, we link out to the appropriate sources that we got these inspirations for these molecules from. So I think that's a JMC article there for JNE 6893, Genentech's HPK1 inhibitor. And so Lou and Shaima did a really amazing job going over exactly what our case studies were, what our brief articles look like. And so these molecule roundup highlights that we present here in these molecules of the month articles, these are truncated versions of both of them, but you can expect to get the same information out of these truncated versions. And so we give you exactly how the molecules were discovered. And so, for example, Genentech's HPK1 inhibitor was discovered through a fragment-based screening and structure-based drug design. We give you a feel for the competitive landscape. And so, of course, HPK1 is a super hot area oncology target right now. And so we list off all of the appropriate companies that are actively engaged. Actually, this list is too long, so that we couldn't even include all of the companies there. But for example, Nimbus is also actively engaging in HPK1, and so they actually presented their discovery story, I believe, at the ACS meeting in Denver. And so we actually have a case study on that. And so if Dennis clicks on that, that will actually take you to the case study for that particular HPK1 inhibitor from Nimbus. And Lou walked us through exactly what these case studies look like. We also provide, a context. Okay, why is this particular HPK1 molecule or particular inhibitor interesting? And it turns out that Genentech found this really clever way of taking advantages of this bell-shaped pharmacology that many companies have noted, and they were actually able to use this bell-shaped pharmacology to really drive their selectivity assays and to optimize kinase selectivity. And so it's a really fascinating story. And this is really one of the reasons why this was selected as a molecule of the month. And so we have 10 molecules every month that we highlight, and we can continue to scroll down. But not only are the success stories highlighted here, but for example, compound 9, Fulcrum Therapeutics, muscular dystrophy, phase 3 candidate was also highlighted. And of course, this molecule failed its pivotal phase 3 trial. And so we also, like to point these things out. Okay, what else beyond the success stories or the discovery stories are out there that the drug discovery community might be interested in? And of course, what we're trying to highlight here are really the transferable insights. What can other companies learn from these programs? What's also super exciting about the molecules of the month and what makes it particularly fun, I think, is we get active feedback from our reviewer community. And so, for example, if we scroll down here a little bit, some of our reviewers get very excited by the molecules we select. And so Charles was clearly very excited by Genentech's HPK1 inhibitor. And so he put together some nice commentary on why he thought this program was particularly notable. And Julian Lefranc at Merck also gave us some nice feedback on why he thinks this program was particularly notable and worth keeping an eye on. So if we then select on this curated molecule database search, so this is very similar to the patent database search. You can actually track, what is going on or what has gone on in the space over the past year or 2 years, for example. I think we go back a few years for this database, but you can of course search. It's very similar to the patent search. You can search the mechanism of action, the originators, the companies that are involved, the stage of development, the therapeutic areas, and when those molecules were featured in our molecule of the month series. And so Dennis is really nicely highlighting that. We had these various filters on the left-hand side there that you can filter off if you're interested in, keeping track of what Genentech is doing. You can filter by company and see what molecules they have published on recently. And so this gives you, again, the ability to track trends in the industry, what companies are doing, and where they're perhaps moving in the future. So this is a really awesome tool to use to keep track of things and to get more inspiration for your programs. So if we just go back to that landing page then, Dennis, or the September 2024 Molecule of the Month article, I guess the last thing I just wanted to highlight here, we do have nomination forms. And so if a molecule really inspires you, you can actually submit a molecule nomination. If you want to become a reviewer, you can also become a reviewer and fill out the very quick application form, and we can take a look, and you can help us select our top 10 molecules. So if we go then back to the landing page for the molecules of the month and scroll down a little bit, we actually walk you through exactly what our process is for how we choose our top 10 molecules of the month. And so we really start off really at the beginning of each month, collecting and collating all of the research that's being published on small molecule drug discovery. And so we pull information from peer-reviewed literature, we pull information from news releases, company disclosures, poster presentations that companies publish on, news releases. And so we're pulling information from every area of the drug discovery universe. From there, we as a team with our backgrounds, we take it upon ourselves to vote for our top 10. And so we kind of have a very democratic system here where we all choose our top 10. It actually turns out, though, that pretty regularly, for example, our top 4 molecules for everyone on the team usually tend to be very consistent. And then there's some variations in maybe some of the other later molecules. And so then we have an open discussion on why we think these molecules are warranted of being noted as a top 10. And then, from that process, we then finalize our top 10, send it out to our reviewer community, and finalize our top 10 and allow the reviewers to provide feedback on our top 10 molecules. Sometimes, for example, reviewers will say, hey, have you thought about this molecule? Did you consider this for your molecule of the month? Oftentimes we have. And then we can kind of have an open dialogue of why we either selected that molecule or didn't select it. And then after that, we will publish our final top 10 molecules like we just did this morning. And then, of course, once we do that, it's out in the public sphere, and we receive feedback from the community of what they think. So, okay, what makes a great molecule? And so this is kind of what we're looking for. So, for example, that's a RMC 6291 there on the left. And so our team is looking for first disclosures of molecules or new properties. We're looking at clinical candidates with interesting profiles, non-clinical molecules with intriguing modes of action, interesting structures or unique properties, drug discovery stories with educational value and transferable insights. That's, of course, always we're always looking for that. And then even nominations from readers and reviewers. And so, again, we're pulling information from all different sorts of angles, but these are really the things that come top of mind when we're looking at molecules and thinking about what molecules we should highlight in the molecules of the month, and then even more generally as a case study or a molecule brief type article. So I think that's what I had, Dennis. So thank you.
Awesome. Yeah, thank you, Rory. And yeah, we oftentimes get asked about how we select those molecules. So we thought we'll let Rory, who's responsible for that process, walk you through it. And yeah, if there are any questions, please let us know. And yeah, I want to conclude it with the structure search now. We also have made really huge enhancements to the structure search recently. So I want to walk you through a few of the new features that are now available. So first of all, I wanted to speak to the different databases that we have. So we have a database of an internal database. We often get asked, how of what types of molecules does this database comprise? And it's, the molecules of the month, as well as the case studies that we have on the websites. Those are fully text and structure searchable as well. Our other database is the FDA approved drug database, and we just added that feature just a few months ago. So I want to demonstrate that. And, if you want to, for example, know a specific, let's say functional group that's present in a drug. Let's say we want to know acetylenes, how many drugs, FDA approved drugs, contain acetylene. So we just draw something in here, that acetylene, toggle down to drugs here, and then search for structure. And we will quickly get an overview of drugs that contain acetylenes. So you can then really quickly see, what is the context in which, those drugs contain acetylenes. And you'll find, oh yeah, it's oftentimes progesterone and estrogen receptor modulators that contain those terminal acetylenes here to protect this alcohol. If that was a secondary alcohol, it'd be metabolically oxidized and deactivated. And that's why it has been found in the 40s that actually those terminal acetylenes are great metabolic protecting groups, if you will. And because these compounds, the progesterone and estrogen receptor modulators, are dosed in so low doses, oftentimes the problems that you have with acetylenes are not necessarily, specifically with terminal acetylenes, for example, time-dependent inhibition are often not a problem. So now let's say, we have this acetylene here, but we want to go back to structure search, and we also want to see, like, not only is there an acetylene in the molecule, but also maybe is there, in addition to that to that acetylene, a cyclopentane in the molecule? And they can be in different parts of the molecule. So let's just see, and you don't have to connect them. You can draw, two different functional groups and then just hit search here as well. And now, whether or not these groups are connected, you will find drugs that contain both acetylenes and cyclopentyl groups, which is really cool. So, you can, like, for example, in a design meeting, if you propose to include, like, say, two different functional groups, you can just basically, do that, search those here and see if they're in drugs as well. And then again, you go back to that page, your drawings are saved, and you can further modulate those kinds of drawings. You can click here to clear the drawing board out as well. And for that FDA-approved drug database, I also want to show that search feature here as well. So if you click here on the FDA-approved drug text search, what you can do is now you can search that database by text. So where that comes in handy is you can, for example, see what were the types of drugs that were approved last year. How did their structure look like? What are some of the mechanisms that are being approved? And, some of the names. So you can really nicely search that by text as well. And then if you have a target in mind, let's say you're interested in SGLT2 inhibitors here, you can also type that in and see what types of SGLT2 inhibitors were approved over the years. And what you will see is shocking, actually. So here, those SGLT2 inhibitors are oftentimes aryl C glycosides. So, you have dapagliflozin here. In the same year, you got empagliflozin approved, which is Boehringer Ingelheim's Jardiance, brought in about $10 billion at the top peak sales per year. And, even 10 years later, the innovation lies in the ether group here. So you have the exact same aryl C glycoside, and only the ether functional group here is modified. So those are some of the trends that you can then identify with that tool. But now, of course, the FDA-approved drug database is limited, right? Not all drugs make it to an FDA approval. So we also provide that molecule database. That's our internal database that I was mentioning. So here you can search a variety of different things. So for example, you can search, and drawing is relatively easy. You don't have to go here. You can just hit buttons like N for nitrogen, O for oxygen. You can hit a 1 here for single bond. You can hit 2 for double bond. It's very easy to play around with that. So let's just check what are some of the acetidine amides that are contained in preclinical molecules, clinical candidates, something like that we have case studies for. And what you'll find is immediately relevant results here. So if you were to do a search like that on SciFinder, you'd likely find tens of thousands of results of maybe how to make acetidine amides, but not necessarily focused on drug discovery and not necessarily focused on why did they even use those acetidine amides? What was some of the context? Like, what impact did it have on the properties? So here on Drug Hunter, we prioritize those case studies where we have additional information, and Lou and Shaima very nicely highlighted what we have. And in those texts, we then, explain to you what, kind of context and what kind of context were those functional groups used, and can you maybe employ them in your designs? Like, would that make sense for something that, you're currently looking at for your program? So, that's a cool feature. And again, you can also search that molecule database here by text as well. So if you're interested now, again, the set of FDA-approved drugs is limited. If you're interested in like a recently hot topic, has been PI3 kinase inhibitors. If you're interested, let's say, in PI3 kinase inhibitors, where we have case studies, you can just type in that mechanism there and, again, find immediately relevant case studies that you then can click into and get directly transferred to those case studies where you can get all these transferable insights. So yeah, one of the things that we've recently been asked is, can you actually copy things, from Smiles or ChemDraw? And the answer is yes, you can. And I want to show how that is done. So with our first disclosure hit list here, we actually just recently also made the INN list searchable. So where that comes in handy is you can find really interesting structural features that are being disclosed in this INN list before maybe being publicly disclosed. And we offer the smile string here. So you can just go there, copy, or, you can take a ChemDraw and, copy the smile string. You can just hit Control-Alt-Copy on your keyboard and then copy this thing here. So basically now I just basically copied the structure here, or you can copy it here in the smiles string. And now you have that structure here in that box, and you can further play with it. You can search for this, or if you say, you're more interested in like a sub part of this, you don't want to search through like for the entire structure. You're more interested in, let's say, for example, this sub part of the molecule, and you want to see kind of what, what compounds are out there that are, maybe drugs that contain this particular motif. And then, you just hit search and you find a BCL inhibitor venetoclax, right? Or if you want to go back and then search our molecule database with that, the same thing, you find other BCL2 inhibitors. So you really quickly can see that this is a very common motif in, BCL2 inhibitors. And yeah, now, of course, the audience may now wonder, my goodness, aryl nitro groups here, that looks like a structural alert to me. What does that even do? So you can probably click in one of our case studies here, Sorontoclax, which we recently discussed, and then go into what Lou discussed, kind of the structural discussion here. So we really discussed here what does this nitro group contribute here. And as you can see, it doesn't make any direct interaction, this nitro group, but it strengthens this pi-pi interactions with this tyrosine 202 here in BCL2. And another thing that you can see with the competitive landscape analysis that we do here, you can see other, again, BCL2 inhibitors that were indexed. This nitro group is often replaced by other electron withdrawing groups. For example, this trifluol group here in navitoclax is used, and also in pelicidoclax. So you see that this nitro group is being replaceable, and that it really doesn't make any direct interactions, but just strengthens this pi-pi interaction. Yeah, so those are a few of the things you can now do with Drug Hunter Structure Search. And again, we're adding new features on almost like a almost weekly basis. So it's really a fast developing tool. Another thing that you can do is, if you want to just type in here, you can just start typing. You can hit P, it's phosphorus, but you can also type, for example, piperidine or piperazine. And you can quickly draw things here. Let's say, for example, you can hit T, then you go through this list in the bottom, select the cyclopropyl. You can hit 2 here, for example, then you get a double bond attached to this. You can hit 1, you get a single bond, 2, a double bond. So it's really easy to navigate with the oxygen here, and then you can search for those types of things, or 2 things in the box, and really quickly find molecules or drugs with that particular structural motif that are approved. So here you can see, for example, a lot of DNA gyrase and topoisomerase inhibitors contain this particular cyclopropyl protected quinolone type of nitrogen. Yeah, and that really concludes what we wanted to share with you today. I think we have a little bit of time for the Q&A. So if there are any questions, please feel free to drop them in the chat or the Q&A box. And again, yeah, the entire team is open now for questions if there are some. And I actually do see some already. So let's go through those. So this is for the entire audience. In the future, is Drug Hunter thinking, and this question comes from Gagan, is Drug Hunter thinking to enable the report to be imported as a PowerPoint? That could be quite useful for making a quick and smart presentation. Secondly, how about having the possibility to have an audible version of the report? This could be super useful as well. Does anybody want to chime in there, or should I take a first stab at the answer? I can answer that, no problem. So yeah, Gagan, thanks for the question. So we actually have been thinking about doing a podcast, so getting these reports into audible formats. And as you know, we oftentimes do these coffee chats where we walk through those reports. It's just currently a matter of prioritization for the team. So with the small but mighty team, we can't do everything. So currently we're really focused on the reports writing. And then oftentimes they're visual learners as well. So oftentimes it's difficult to visualize those structures, right? And it's difficult to put that into an audio podcast. But short answer is we have been thinking about it. And for the PowerPoint export, so currently, we basically, don't allow these databases to be exported. But for PowerPoint reports, we actually moved away from like PDFs. Our molecules of the month used to be just, or the Drug Hunter in general used to be just monthly PDFs, and we find that a interactable format with links that people can interact with is a little bit more conducive to our readers. Yeah, we have a second question here as well. Can Drug Hunter perform a customized search for a subscriber on a known molecule or a chemical landscape that's not listed on Drug Hunter? Anybody wants to chime in there?
So we have had this suggestion from a couple of subscribers actually. Someone described Drug Hunter as a information CRO, right? And it was much in the same way that you may not have time to make all of the analogs of a compound you might want. You go to a CRO and say, okay, can you go and do this? And we've had some people ask that. I think part of the value of Drug Hunter, like you said, we have like a small but mighty team is, we need to be able to publish all the research we do. And I think a lot of companies would want, if they want us to go in depth on a target, they'd want to have that just between us and them. And I think Drug Hunter works because we're able to share it with our whole community of members. So I think if people were willing to have that shared, I mean that's certainly something we could do. But people are welcome to suggest molecules that they want us to cover, right? If drug discoverers out there in the community think that it's important or it's interesting, that's the kind of thing that we'd want to cover, right? So we certainly take that feedback and really, want to incorporate that into how we cover different molecules.
Yeah, and I'll just add on to what Matt just said. To maintain both the perception and the reality of our editorial independence, we do not currently engage in any sort of consulting activities. So that's really important to point out. And there are a lot of services out there for companies when they want to do a deep dive in a certain area. That's available. And so, at this point at Drug Hunter, that's not a service that we envision providing at this time.
Yeah. Awesome. Let me see there. I don't see any further questions, and maybe that's actually great because we're coming up on time. So it's now 10 a.m. Pacific and I think 1 p.m. Eastern. So yeah, I want to thank everybody on the panel today. Really a nice job in giving a great overview of Drug Hunter to our members and prospective members. So very, very nice. Thank you to the audience. Yeah, thanks for the questions. And if there's anything open that we weren't able to answer today or that, didn't come to mind asking today, please feel free to reach out to us anytime. Also, if you're watching this video on YouTube, feel free to reach out to us if you have any questions and didn't get the chance to attend live. And with that, we'd love to see you at our next event, which is going to be a flash talk next Thursday with the title Advancing Macrocycles in Drug Discovery. So a really interesting topic recently, and it's being presented by Kadlev Terrace, former CSO of Velia Therapeutics. So a very interesting speaker, an expert in the field who has been working in the field for more than 25 years. So I would highly encourage you to dial in. And then on December 5th, we have our very own Lou Pennington actually presenting a flash talk as well. And I would highly encourage you to register for that flash talk as well. All right. So with that, happy Friday, everyone. And we'll see you at the next event. Take care. Bye-bye.
Thanks a lot.
Take care.